You know, you probably remember the golden rule of your high school biology class.
Oh, yeah. The powerhouse of the cell.
Exactly. Even if you forgot literally everything else, you remember that the mitochondrion is the powerhouse of the cell.
Burned into all of our brains.
Right. We've all seen those neat, color-coded textbook diagrams, you know? There is the nucleus sitting right in the middle, acting as the command center.
Yeah, and floating somewhere off in the cytoplasmic goo around it are the mitochondria.
Just these little free-floating batteries. We were taught to picture them just churning out energy and releasing it into the watery void of the cell.
Just hoping it drifts to whatever needs it.
Yeah, exactly. It's a very clean, sort of chaotic picture.
Yeah.
But what if everything we've pictured about cellular architecture is wrong?
Which is exactly what we are finding out.
Right. What if that battery isn't just floating around at random? Imagine, if you will, that battery is actually physically hardwired, plunging a dedicated power cord directly into the cell's command center.
Like a plug into a wall outlet.
Yes, a direct physical tether to the nucleus.
I mean, it completely upends how we understand cellular logistics. We've known for a long time that different compartments in a cell communicate. But a hardwired physical energy grid existing at a microscopic level.
It's mind-blowing.
It really is. It's a revolutionary shift in how we view the basic building blocks of life.
So today we are doing a deep dive into a truly groundbreaking 2026 scientific paper. This research proves, for the very first time, the existence of this direct physical tether between mitochondria and the nuclear pore complex.
And the amount of evidence they gathered is staggering.
Oh, it's a massive stack of evidence. We are talking high-resolution electron microscopy, advanced AI protein modeling, CRISPR gene editing, and even in vivo live mouse models.
They really covered every base.
They really did. And our mission today is to figure out not just what this microscopic power cord looks like, but how it actually works. So let's start with the visual evidence.
A place to start.
Because, I mean, if my textbook was wrong and these things are actually touching, how did researchers even spot this microscopic tether in the first place?
Well, they had to look much closer than anyone really had before. They used something called transmission electron microscopy, or TEM.
Break that down for us.
Sure. Think of TEM as taking the ultimate zoom lens to a cell. Instead of using light, it shoots a beam of electrons through a sample.
Wow. Okay, so electrons instead of light?
Yeah, because it allows you to see structures at an almost atomic scale.
Yeah.
So they looked at several types of tissues: heart tissue, brain tissue, and a specific connective tissue cell line called 10T12.
Heart and brain. Okay, that feels important. We'll definitely come back to that.
Definitely. So under that intense magnification, they saw something striking. A subset of mitochondria were sitting right up flush against the outer membrane of the nucleus.
But wait, I've seen electron microscope images. Everything just looks like dense gray blobs and squiggly lines, right?
Yeah, it can be pretty messy.
So at that level of magnification, how do the researchers know for sure they are looking at a mitochondrion and a nucleus rather than just, random cellular junk?
That is where the biochemical markers come in. They essentially painted specific proteins with fluorescent tags so they would light up.
Like putting a neon sign on a specific building.
Exactly. First, they tagged a protein called SYNE3. Now, we know SYNE3 is a structural component that only exists on the outer membrane of the nucleus.
Okay, so that establishes the wall of the command center.
Right. And sure enough, the mitochondria were nestled right up against the SYNE3 markers. But the researchers went deeper. They tagged another protein called NUP107.
And what does NUP107 do?
Well, NUP107 is a core building block of the nuclear pore complex.
The doorway.
Exactly. The nuclear pore is the actual doorway into the nucleus. It's the heavily guarded gate that lets instructions out and materials in. The imagery clearly showed the mitochondria weren't just touching the nucleus.
They were associating directly with these specific doorways.
Yes. The microscopy revealed dense, tiny, filament-like structures physically bridging the gap between the surface of the mitochondrion and the nuclear pore.
Okay, let's unpack this for a second. Think of the inside of a cell like a crowded, thick swamp.
That is an accurate description.
There is a lot of traffic. So imagine you are looking at satellite imagery of two massive corporate buildings in a crowded city. From a distance, you assume they are completely separate.
Right.
But you get a better camera, you zoom way in, and you realize there is a dedicated, enclosed sky bridge physically connecting them. That's what the microscopy gives us.
That's a great analogy.
But I have a skeptical question, and I'm sure the researchers had it too. The inside of a cell is packed. Things must bump into each other constantly, right?
Oh, all the time.
So how do they know the skybrin is a permanent structural part of the architecture and not just a temporary microscopic traffic jam?
That is, the researchers used proteomic screens, basically putting the mitochondria through a molecular sorting machine to see exactly what proteins were stuck to them.
Like tearing down the bridge and cataloging every single brick.
Exactly. They purified intact mitochondria and ran them through a mass spectrometer to identify every single component. And stuck to the outside of these purified mitochondria, they found proteins that belong exclusively to the nuclear pore complex.
Wow. So they pulled away the mitochondria and found pieces of the nuclear doorway coming with them.
Precisely. And the most abundant one they found was a protein called RANBP2.
RANBP2.
Yeah. Now, RANBP2 is essentially the structural tentacle of the nuclear pore. It makes up the cytoplasmic filaments, which are the parts of the pore that reach out from the nucleus into the surrounding cellular fluid.
Okay, so that's the hand reaching out from the nucleus.
Right. But to really prove this wasn't an accident, they had to test the dependency of the connection.
Wait, how do you test that?
Well, you break something on purpose.
Ah, classic biology.
Yep. The researchers genetically modified some cells to lack certain internal components of the mitochondria. Specifically, they broke complex I, or complex 3, of the electron transport chain.
Which is the internal engine that generates the power.
Exactly. When they broke the internal engine of the mitochondrion, the amount of RNBP2 attached to the outside drastically dropped.
Oh, wow. That is fascinating. So the bridge isn't just a static piece of tape. If you break the generator inside the mitochondrion, the nucleus literally lets go of it.
Exactly. It proved that this physical association is an active, dependent connection. The mitochondria and the nuclear pore complex were deliberately holding hands, and RNBP2 was the hand reaching out from the nucleus.
Okay, so we have our bridge, and we know RNBP2 is the hand reaching out from the nucleus. But what is reaching back? Like, what is the specific molecular brick on the mitochondrion that RNBP2 is grabbing onto?
So to figure that out, the researchers deployed advanced proximity labeling tools, techniques called GST pull-down and BioID.
BioID, okay. How do those work?
You can think of these as chemical traps. You attach a molecular bait to RNBP2, and it chemically tags whatever protein it physically touches.
Oh, that's clever.
Right. So when they ran this trap, the top mitochondrial candidate that emerged was a protein called VDAC1.
VDAC1. Break that down for us. What is its normal day job?
VDAC1 is an outer mitochondrial membrane protein. It is essentially the primary channel, or pore, on the surface of the mitochondria.
So it's the main exit door for the energy molecules a mitochondrion produces.
Exactly. And the chemical traps prove that VDAC1 binds directly to a highly specific section of RANBP2 known as the C-terminal domain, or CTD for short.
And the paper notes something really interesting about this specific C-terminal domain. It is incredibly protected by evolution. They call it vertebrate-specific with very low genetic divergence.
Right.
Which means from mice to humans, evolution found a design for this specific docking station millions of years ago, realized it was absolutely essential for survival, and just locked it in. It hasn't mutated or changed because it works perfectly.
Yeah, that evolutionary lock is exactly what allowed the researchers to map the structure so beautifully. They used AlphaFold for this part.
The advanced AI system that predicts how proteins fold into 3D shapes.
Yes, they used it to map the exact interaction between the two proteins. The AI showed that these proteins aren't just bumping into each other. They have specific chemical teeth that lock together perfectly to create a seamless, watertight seal.
Chemical teeth, like down to the specific amino acids.
Down to the exact residues. On the RANBP2 side, it's the amino acids phenylalanine at position 2977 and aspartic acid at 2978.
So F2977 and D2978.
Right, and they lock right into glutamic acid and two threonines on VDAC1, positions 50, 51, and 52.
That is absurdly specific. But no, wait, let me push back on this for a second.
Go for it.
If VDAC1 is the main exit door on the mitochondrion, and you plug RANBP2 directly into it, doesn't that block the hole? I mean, think about it. If you plug a thick, heavy cord into a wall socket, you completely occlude the socket. Nothing else can get in or out. So does attaching this nuclear tentacle plug up the mitochondrial pore?
That is a great question. And what's fascinating here is the elegant geometry of the connection. AlphaFold showed us that the binding actually does not occlude the VDAC1 pore.
It doesn't.
No. Instead of a plug going into a drain, think of it more like two pipes fusing together. It creates an extended continuous pipeline, And the diameter of this new pipeline is roughly 10 angstroms.
10 angstroms. To give a visual for that, an angstrom is 1/10,000,000th of a meter. We are talking about a pipe so unfathomably tiny that it's measured at the scale of individual atoms.
Yeah, and that 10 angstrom size is no accident. The researchers found it is the exact perfect size to allow specific high-energy molecules to slip right through from the mitochondrion directly into the nuclear pore.
And the VIP molecule they focused on was phosphocreatine, right?
Correct. Phosphocreatine, or PCR, is a critical, highly dense energy carrier. Its physical diameter is about 6.8 to 8.2 angstroms.
Oh, wow.
So it fits right through that 10 angstrom pipe with just a tiny bit of room to spare, like a bullet traveling down a perfectly machined barrel.
I love that image. Okay, let's do a quick recap here just to cement this visual in our minds because we've covered a lot of ground.
Yeah, let's recap.
So we started with the old textbook model, mitochondria floating around randomly in the cellular goo. But now, zooming way in, we've found a physical structural skybridge. We know the exact building blocks. VDAC1 on the mitochondrion physically locks together with the C-terminal domain of RANBP2 on the nuclear doorway.
Perfectly locked.
And instead of blocking each other, they fuse to form a perfectly sized 10 angstrom pipeline designed specifically to shoot dense energy molecules like phosphocreatine straight into the command center.
That covers the structural reality perfectly. But in biology, structure is really only half the story. The next logical question is function.
Right. Building a dedicated pipeline takes a lot of cellular effort. Why does the cell go through the trouble?
Exactly. So what does this all mean for the cell? To figure out the why, the scientists did what scientists do best. They destroyed the bridge to see what would break.
Oh boy, how did they do that?
They used CRISPR, which acts like highly targeted molecular scissors, to genetically engineer a cell line where they deleted RANBP2. They essentially wiped the bridge out of existence in vitro.
And what happened?
Physically, the first thing they observed was the mitochondria drinking away from the nucleus. Without the tether, they just floated back into the cytoplasm.
Back into the sun.
Back into the swamp. But the biochemical result of that drift was absolutely staggering. They wanted to measure the actual energy levels inside the nucleus, so they used a specialized biosensor.
What does that do?
It's essentially a molecular alarm system injected into the nucleus that literally glows under a microscope when it detects ATP.
And ATP is the cell's basic energy currency.
Right. So in the cells where the bridge was cut, that glow almost vanished. They recorded a massive, crippling drop in nuclear ATP and phosphocreatine levels.
Let's use an analogy to really understand why this drop happens. Let's go back to that idea of the cytoplasm being a thick, crowded swamp.
Okay.
You can have a massive state-of-the-art power plant, which is your mitochondrion. But if you have to carry high-voltage batteries from that power plant by hand, trudging through miles of thick, muddy swamp without a dedicated, insulated copper wire, you are going to lose most of your charge before you ever reach your destination.
That is incredibly accurate. And the researchers actually mapped this out biophysically. They created a mathematical model to look at how ATP diffuses or drifts through the cell.
Because it's not a clear path, right?
Not at all. They had to account for the physical resistance of the crowded cytoplasm and the fact that thousands of other enzymes are lurking in that swamp, actively stealing and consuming ATP as it floats by.
So it's a gauntlet.
Yes. Their model showed that if a mitochondrion moves from being physically tethered, which is just 50 nanometers away from the nuclear pore,
Okay, 50 nanometers.
to being untethered, say 500 nanometers away.
Which is just a tiny microscopic drift in the grand scheme of things.
It is, but the energy concentration actually hitting the nuclear pore drops by a staggering 90%.
90%. You lose almost all your power just from cutting the cord and forcing the energy to drift through the swamp.
Yeah, and the cell simply cannot afford a 90% power loss because the nucleus is incredibly power hungry. Think about what the nucleus actually does.
It houses the DNA.
Exactly. It needs massive continuous surges of energy to drive processes like DNA synthesis, identifying and repairing genetic mutations, and physically unpacking the dense chromatin structure so genes can be read.
And when the researchers looked at the cells with the broken tether,
They saw them struggling to survive. Slower growth across the board, downregulation of histone modification, and transcription pathways.
the paper specifically notes they suffered a defect trying to transition from the G1 phase to the S phase of the cell cycle.
Right, which is huge.
For those who aren't biology nerds, the S phase is the synthesis phase. It's the moment the cell decides to unzip and copy its entire genome.
which requires an astronomical amount of energy.
Right, and the cells with the broken bridge just didn't have the juice to copy their DNA. Crucial pathways were just shutting down.
And to fully close the loop on this system, the researchers pointed out a specific enzyme waiting inside the nucleus called nuclear creatine kinase, or CKB.
Why is CKB important?
Because phosphocreatine, that dense energy molecule we talked about traveling through the 10-angstrom pipe, it cannot be used directly by most nuclear machinery.
Oh, I can't.
No, it has to be unpacked and converted back into ATP. CKB is the unpacking machine.
Oh, I see.
The fact that the nucleus is purposely packed with CKB proves that it is specifically designed to receive and process the shipments coming through this physical bridge.
You have the generator on one side, the 10-angstrom power line in the middle, and the unpacking station waiting right on the other side.
It's a completely closed, perfectly engineered circuit.
That is just, it's amazing. But, it's one thing to see an isolated cell in a petri dish struggle. When you cut this circuit, it begs a much bigger question.
What happens in a whole animal?
Exactly. If a single, lonely cell can't function without this bridge, what happens to a living, breathing animal if this connection never forms?
This is where the researchers escalated to the macro level using in vivo mouse models.
Okay, live mice, what did they do?
They genetically engineered mice to lack just the specific C-terminal domain of RANBP2.
Just the docking station.
Right. They didn't delete the entire nuclear pore, which would obviously kill the cell instantly. They just removed that tiny docking station that the mitochondria plug into.
And here's where it gets really interesting, folks. When they deleted just that specific microscopic docking domain, the mice did not survive development. It resulted in embryonic lethality.
They simply could not develop. And when they investigated why the embryos were failing, they found severe defects in very specific developing tissues.
Which tissue?
Primarily the developing heart and the neural crest.
And the neural crest is the group of embryonic cells that eventually forms the brain and the nervous system. It is absolutely no coincidence that the heart and the brain are notoriously the most energy-demanding, power-hungry tissues in the entire vertebrate body.
None at all. If we connect this to the bigger picture, the biological logic is stunning. Think about cellular differentiation.
Okay.
That is the process where a generic, blank slate embryonic cell transforms into a highly specialized beating heart muscle cell, or a complex neuron.
It's a total identity change.
And that transformation is arguably the most exhausting thing a cell will ever do. It requires massive, whole-scale restructuring of the DNA and chromatin within the nucleus. You're essentially tearing down and rebuilding a skyscraper from the inside out.
And you can't fuel a massive construction project by throwing batteries through a swamp.
Exactly. Without that direct physical energy bridge, the developing heart and brain cells simply cannot get enough power into the nucleus fast enough through basic diffusion.
They just run out of gas.
The paper notes that without the tether, these cells remain stuck in a primitive, fast-cycling state. They simply cannot mature. By docking mitochondria right at the nuclear doorway, the cell entirely bypasses the slow diffusion of ATP through crowded spaces. That microscopic power cord is what allows the rapid, life-sustaining development of our most vital organs to occur.
It is honestly mind-blowing. Let's trace the full journey we just took today because it scales in such an incredible way.
It really does.
We started at the absolute smallest scale imaginable, a 10-angstrom pipeline built by two proteins, VDAC1 and RANBP2, locking together like perfectly machined puzzle pieces.
The chemical teeth.
Right. We saw how that tiny connection solves a massive 90% energy loss problem for the cell by bypassing the swamp of the cytoplasm. And then we scaled all the way up to realize that this invisible microscopic power cord is quite literally what allows a heart to fully form and start beating in a living organism.
It forces us to completely rethink cellular logistics. And you remember earlier we discussed how that RANBP2 docking station is vertebrate-specific and highly conserved by evolution.
Yeah, that evolution locked it in millions of years ago.
That tells us a fascinating story about our own history. As life got more complex, as animals evolved high energy demands for large, complex brains and highly efficient cardiovascular systems, we couldn't rely on free-floating batteries anymore.
The old diffusion model just wasn't good enough to power a human brain.
Exactly. Evolution had to invent a hardwired power grid to make human life possible.
Which leaves us with a truly fascinating and maybe slightly unsettling question to ponder as we wrap up today's deep dive.
Uh-oh. Let's hear it.
Well, if our cells evolved this highly specific physical power cord just to fuel the intense energy demands of the nucleus, What happens when that system is hacked or breaks down?
Oh, yeah. That raises a massive question about aging and disease. Think about the aging process. Over time, our cellular machinery naturally degrades.
Right.
Do these physical tethers between the mitochondria and the nucleus start to fray as we get older? If the bridge weakens, the nucleus is forced to rely on diffusion again, losing 90% of its power. Is that energy starvation part of why our cells lose their ability to repair their own DNA as we age?
That is a chilling thought. Or look at it from the darker side of biology. Cancer.
Yeah.
Cancer cells are infamous for their chaotic, massive energy demands. They have to constantly rapidly alter and copy their DNA to grow and evade medical treatment.
Which requires a ton of ATP hitting the nucleus.
Exactly. Could cancer cells be actively hijacking this exact power grid? What if a tumor is just a cluster of cells that has figured out how to purposefully build thousands of extra VDC1 RANBP2 tethers, permanently hardwiring their engines to fuel their own unchecked growth?
It is a terrifying thought, but it is the exact next frontier for this research.
Yeah.
Now that we know this bridge exists, we have to see who else is using it. If cancer relies on this tether to survive, then maybe the cure lies in finding a highly targeted drug that acts as molecular scissors.
Cutting the cord and starving the cancer's command center without harming the rest of the cell.
Precisely.
It is a completely new paradigm for looking at the biology keeping us alive. Well, I want to thank you for joining us on this deep dive today.
It was a pleasure.
And to you listening, the next time you picture a cell, I hope you don't just see a command center and some floating kidney beans drifting around in a watery void. I hope you visualize a bustling city fully hardwired, pumping energy through unfathomably small pipelines to keep the whole operation running. Keep questioning the invisible machinery out there, and we'll catch you on the next deep dive.